Hello everyone, my name is Eman Sharafeldeen Yusuf. I am a researcher and a part-timer teaching assistant at Department of Pharmaceutics, Faculty of Pharmacy, University of Khartoum. I am very happy to participate in the Global Conference of Nanomedicine, Nanobiology, Nanotechnology and Pharmacology, and I am honored to share my research with you.
Mangifera indica, the mango tree, has been known by its sweet fruit, and in domestic medicine, the tree has been used as therapy against chronic dysentery, also in the prevention of cancer, scabies, asthma, and bacteria. Well, today I'm going to talk about Mangifera indica from another scientific point of view, which is extraction and evaluation of Mangifera indica gum as a sustained-release polymer in glibenclamide matrix tablet. First, I'm going to talk about the drug delivery systems. Depending on the mechanism of the drug release, there are two classes: either immediate drug delivery systems or modified release drug delivery systems. The modification means application of change to already existing drug. This done in order to improve stability, efficacy, drug safety, or the patient compliance. Modification of drug release is usually achieved by the use of barriers. One of those barriers is the polymeric materials, and they are usually inert, hydrophobic, or hydrophilic in nature, with the drug either dispersed in the polymer matrix or enclosed in the polymer membrane. Polymers are natural, synthetic, or semi-synthetic ones, but the natural polymers are preferred. Why? Because of some feature that they possess. Natural polymers lacking toxicity, comes with low cost, has a soothing action and non-irritant nature. They are biocompatible and they are abundant in nature. Well, one of those natural polymers is the Mangifera indica gum. Mangifera indica gum belongs to the genus Mangifera of the family Anacardiaceae. It is the dry gummy oxidate polysaccharides obtained from the bark of the Mangifera indica plant. This resin is poured outside the mango tree's trunk by the act of wind, fire, or any small injury on the trunk surface. After that, those gum resin is collected and then purified to be used in pharmaceutical manufacturing. Studies were performed on this gum for its binding, sustained release, and disintegrating properties. Now, let's come to our main objective on this research. Why modified release systems? Why we are making such an effort? Why we need them? We all know that the ideal drug delivery system is a system that delivers the drug to its specified site of action for the required period of time, with its concentration kept within the therapeutic range in order to produce maximum therapeutic response. Well, this can be achieved by conventional dosage form, right? But in this case, it will be just for a short period of time. Besides, it will require frequent administration to maintain the level within the therapeutic range, and that may eventually lead to peaks and troughs in the plasma concentration curve with increased drug toxicity and reduced compliance. Therefore, modified release systems give us a solution to those problems. They ensure that the drug is released over a prolonged period of time at specific site of action in a desired concentration within the therapeutic range. This will lead us to the next question: why we use natural polymers instead of synthetic and semi-synthetic ones? Natural polymers, as we mentioned before, possess features that enable them to compete with the synthetic polymers available in the market. These features have enabled them to meet the request of the drug delivery systems. And Mangifera indica is one of the plants from which these polymers come from. Therefore, the objective of this research is to extract and evaluate a pharmaceutical-grade Mangifera indica gum as an excipient in sustained-release oral dosage formulation using glibenclamide as a drug. So, The general and specific objective will be: first, extraction and characterization of the Mangifera indica gum, then preparation of the matrix tablets of glibenclamide, and finally evaluation of the Mangifera indica gum as a matrix former. The material of this research include: first, Mangifera indica plant, which collected by inside the trunk of the Mangifera indica plant in الفكهاشي من الخرطوم بحري. Then the glibenclamide is a gift sample from Blue Nile Pharmaceutical Industry, Sudan, and the other excipient we get them from Omdurman Islamic University Pharmaceutical Lab, which, by the way, a major part of the research was carried out on it. Now, let me explain how we did it. This included 3 steps. The first step was extraction and evaluation of Mangifera indica gum. The gum resin was collected from the Mangifera indica trees by injuring the trunk side. It was dried, grounded, and passed through sieve number 80. The dried gum, which is about 85 grams, was stirred in 500 ml distilled water for 8 hours at room temperature. The supernatant was obtained by centrifugation. Then the residue was washed with water, and the washing were added to a separate supernatant, and the procedure was repeated 4 more times. Finally, the supernatant was made up to 500 ml and treated with twice the volume of acetone by continuous stirring. The precipitated material was washed with distilled water and dried at 55 degrees centigrade under vacuum. Finally, the dried gum was pulverized and stored in tightly closed container. The second step was physiochemical and phytochemical characterization of the gum. The physiochemical properties, such as organoleptic properties, solubility, swelling index, pH determination, loss on drying, bulk density, tap density, and angle of repose, were all determined according to official procedures. On the other hand, preliminary tests were performed to confirm the nature of gum obtained. The chemical tests that were conducted are ruthenium rate test, Molish test, test for reducing sugar, and Wagner test. The third step was preparation and evaluation of the matrix tablet. The matrix tablets were prepared by wet granulation method. Glibenclamide, Mangifera indica gum, and microcrystalline cellulose were all mixed together, and the mixture was passed through mesh number 60. Granulation was done using a sufficient isopropyl alcohol. The wet mass then passed through mesh number 30, and the wet granules were air dried in the oven. The granules were then sized with mesh number 35 and mixed with the magnesium stearate. The tablets are finally prepared by compressing them using single rotatory tablet press machine. 4 different formulas having different concentration of Mangifera indica gum were prepared. This table showing Formula 1 composition, which contain 1% of Mangifera indica gum, and the image beside the table showing the appearance of the tablet. The second table showing Formula 2 composition, which contain 30% of Mangifera indica gum, and the image showing the appearance of the formula tablet. The third table showing Formula 3 composition, which contain 40% of Mangifera indica gum, and the image showing the appearance of the tablet. You can notice that the color of the tablet become more brownish as we increase the percent of the Mangifera indica gum. The last table showing the composition of Formula 4, which contain 50% of Mangifera indica gum, which is the highest percent among all the formulas, and the image beside it showing the tablet of the formula appearance. The table in front of you is showing the physiochemical characterization results of Mangifera indica gum. As you can see, the average yield of dried gum obtained from Mangifera indica trees was 31% weight per weight, and the extracted gum was brown color powder. This powder was found to be slightly soluble in water and virtually insoluble in acetone and ethanol. Also, this powder is tasteless with no characteristic odor. The second part of the table showing the rest of the results. You can notice that the swallowing characteristic of the Mangifera indica gum was studied using phosphate buffer with pH 7.4 and water. The swallowing ratio was determined as 15 for water and 10 for phosphate buffer. Generally, results showing that Mangifera indica gum has high swallowing index, suggesting that the gum may perform as well as binder, disintegrant, and matrix former. And the reading of the pH was 7.35. The compressibility index, Hausner ratio, angle of repose of Mangifera indica gum were 22.5%, 1.3, and 26.5 degree respectively, implying that the Mangifera indica gum has a good flow with moderate compressibility. This image is showing the Mangifera indica gum powder that has been used in the preparation of the glibenclamide matrix tablet. This table is showing the results of phytochemical screening of Mangifera indica gum. Phytochemical tests that done on Mangifera indica gum assured that the gum lacks alkaloids and tannins, and when the mucilate was mixed with ruthenium red, it gave red color, which affirmed the presence of the mucilate. Also, in reaction with Molish reagent, a violet ring was formed, confirming the presence of carbohydrates. There are no reducing sugar because the mucilate didn't reduce filling solution. The third table is showing the results of the evaluation of the matrix tablets. As the image has shown before, the glibenclamide matrix tablets were baked to brown in color, smooth, and flat-shaped in appearance. The hardness of those matrix tablets was found to be 3.47, 2.91, 2.50, and 2.37 kg per centimeter for F1, F2, F3, and F4 respectively. On the other hand, the friability values were found below 1% for all the formulation. Those results indicating that as the percent of the polymer increase, there is a decrease in the hardness values accordingly. So, why we got such a result of the hardness? Taking in account that the proper die size and the maximum force of compression were used in the process, those hardness results may be because of the absence of the binding agent from the matrix tablet formulation. As we can see, regarding the weight variation and the drug assay, the results were found within the limits. This figure illustrates the swallowing characteristic of Mangifera indica containing tablets. After calculating the swallowing index with respect to time, a direct proportion was found. You can notice that as the time increased, the swallowing index was increased too, and also as the Mangifera indica polymer percent increased, also the swallowing index was increased. The percent weight gained by the tablets was used to estimate the magnitude of swallowing. This was applied for all the formulation in order to study their behavior. When we carried out the dissolution studies on the glibenclamide matrix tablets, we used UV as a detection method. But unfortunately, we discovered that the Mangifera indica gum had the same wavelength range as glibenclamide, which is 213 to 232 nanometer. Due to this interference, HPLC was used as a detection method. After taking several readings, we found that the percent of release is very low, so we extend the profile to 24 hours.
This figure illustrates the results of in vitro dissolution profile of different matrix tablets formulation. The result indicated increase in the release retardant property of the gum from all glibenclamide matrix tablet formulation as the polymer concentration increased. We can notice that there is a sudden decrease in the cumulative percent after the first 2 hours. This sudden decrease may be due to 2 reasons. Either there is an increase in the gel strength and therefore formulation of gel layer is longer path of diffusion compared to the diffusion coefficient of the drug, or the second reason may be that the Mangifera Indica gum polymer relieves the drug as a pulse after a lag time, which means a pulse-style effect. A trial formula of glibenclamide immediate release tablets was prepared to exclude any problems in the dissolution of the glibenclamide that may affect the glibenclamide matrix tablet cumulative percent results. The percent release of the immediate tablets was found to be 45% in 30 minutes. This indicates that the cumulative percent results of the glibenclamide matrix tablets were only due to the sustained effect of the Mangifera Indica gum polymer. In conclusion, the method of extraction and characterization is accurate, reproducible, with low cost, and give high yield. The polymer obtained has high purity, good swelling, good flowability, and the drug release from Mangifera Indica matrix tablets formulation was dependent on the mu-solid to drug ratio, which means as the concentration of the Mangifera Indica gum increase, a drug release is retarded. Our first recommendation is to use Mangifera Indica polymer with very small percent in the formulation, which means less than 1%, or for sustaining period more than 24 hours. The second recommendation is further physiochemical characterization like NMR chromatographic methods that can be performed in order to identify any specific structure of the Mangifera Indica gum polymer. I would like to extend my profound gratitude to my able supervisor Dr. Taib Suleiman Al-Amin Abbas for his support on the manner and how this research work came to be. Sincerely, my limitless gratitude goes to my family and friends for their encouragement. Their immeasurable support is greatly appreciated. We dedicate this work to the martyrs of the glorious December Revolution. They have given us a homeland with their souls. Glory and eternity to them in their high places. I would like to thank the SciTech Central Conference Committee and members for their efforts in this great scientific event and also for inviting me to be part of it. Also, a deep thanks to all of you for listening and following me in this presentation. Have a good day.